Hi, I'm Eileen Furlong. I'm one of the organisers along with Joanna Wasaka and Claude O'Shea of the Keystone Chromatin Architecture meeting in development and disease. The meeting had to be postponed due to COVID, but thankfully it's going to be on in March next year. And we're really excited because, especially because of the pandemic, it's very important to get us all together. And this meeting is going to be particularly special because it's a joint meeting with the Chromatin Epigenetics conference, and there'll be lots of obvious interplays between those two fields. The same proteins are involved, and actually lots of the researchers are working in both of these two fields. So I think there's a fantastic lineup of speakers because of that. I mean, the scope of the meeting is really to show, to try to gather researchers together that are working in first of all on the same theme, which is chromatin architecture, but in different contexts, both in development and disease, but also from different disciplines. So we have people there studying this from a genomics point of view, using chromatin conformation capture techniques, from an imaging point of view, super resolution microscopy, even from an electron microscopy point of view, as well as people using mathematical modeling. So it's to really bring all of these people together to promote collaborations, etc. So it's very exciting. Well, one of the big advances in the field at the moment, in the whole chromatin topology field, is the potential role of phase separation, and also in how our enhancer is communicating with promoters. So that's my own particular interest in enhancer promoter regulation, so I'm a bit biased in that point of view, but we used to think these came in direct proximity to each other. Now it's thought more like that they're in kind of a phase or in a transition, and this is quite exciting now. There's many different alternative models that one could think of, and it would be very important to bring people together to start discussing those and to start to see how can we dissect this. I mean, I'm always interested to hear of new technologies, because new technologies is what brings the field forward. So, I mean, I don't know what's going to be presented. It's going to be in March next year. But yeah, I'm really excited to hear that, to see what new type of technologies are there. How can we look at this in a more dynamic way? How can we look at this at a single base pair resolution? These type of things. Well, I hope the meeting will bring people together. I mean, we haven't been together actually for a long time because of COVID, but also bringing people together from these two different conferences at the same time to promote collaborations, maybe fertilize a bit between people working on human disease versus people working on more fundamental properties of how this is regulated during development. In the end, it's the same common principles that are regulating this, so we all need to start thinking about this and learning from each other's fields. So that's what I would hope we would get. I think the meeting is for people at all stages actually. I mean, for PhD students, postdocs, but also for group leaders, PIs. I mean, this is where you can really promote collaborations. You can showcase your research. If you're a student, you can look for a postdoc position, postdoc faculty position, but also for faculty, like we can form new collaborations, etc. So I think it's for everybody. I think it's a fantastic lineup of speakers, but will select speakers from the abstract. So this is also very important, and we'll try to purposely select junior people from the abstracts, and also people that will really complement the themes of the meeting. So we're trying to sort of showcase a diversity in the field.